sciences. Further to his skills, he has good knowledge of flavonoids, antioxidant activity, oxidative stress, medicinal plants and herbs, animal handling, purification of herbal compounds, bioinformatics, phytochemistry, polyphenols, and free radicals, etc. He has published 28 research articles in journals as author and also as a co author. With this brief introduction, I will welcome Dr. Sushil Kumar Midda for his talk on Introduction to Biostatistics. Sir, over to you.
Yeah, thank you very much, ma'am, for a nice introduction and to call me on this platform where I can interact with the students directly. So, good morning, everyone. All of you are, I think you can hear me. So, please on your browser and write joinpd.com. All of you kindly write joinpd.com. If all of you are able to see my screen, all of you are able to see my screen.
Yes, sir, it's visible.
Yeah, what is written joinpd.com, correct?
Yes, sir.
So kindly write joinpd.com and then write this IQZAZJH. I need minimum one student to join me. So it's good for you if you join me. It's we will let's make the session interactive. Let's go with handy because there is no yeah, thank you very much. My first student has joined. I kindly keeps on joining so I can start the class. Others also please join it. It's good for you, I am telling you again. You will be feeling happy after seeing this all. You can see the slides. You can use this. Kindly write down this code if you are not able to join. Okay. Please write joinpd.com. If you are facing any problem, let me know. Please write in your browser, in Google. Open your browser, write joinpd.com and write this code LQZAJH. Yeah. So shall I start? But till now I have only one student. Out of 70, only one is able to join me. You all are MSc guys, correct? So you should know how to use this all presentation skills. Yeah, another one has joined. Great. Now I have two students, three, four, five, six. Wow, that's great. Happy to hear this. Happy to see. Eight. Wow. So you can see the number is changing, correct? All of you are seeing. All of you are able to see. After 10 students comes, I will start the class. Already 12 are joined me. So let me start the class. So what you are able to see now? Let's be interactive. You can speak with me when whatever questions you have, you can ask me always. Okay. So we are learning biostatistics. Though it is topic is introduction to biostatistics, so I thought I will add it biostatistics and research. Yeah, what is statistics? Let me start with. So it's very tough task to whenever we start and we start learning. People always think statistics as a very very like tough task to work on. But being an MSc students, being a researchers, you need to do it at some or other places. You need it to work on it because without statistics, people may not take your publication, your paper. Publishing paper will become a little tough task. So let me. So as you are able to see, I am Dr. Sushil Kumar Midda. I am a coordinator in MLSC, Mahalakshmi Amani College for Women, Bengaluru. I am starting today. And thank you to Dr. Anuradha. Anuradha madam is one of my uh, guide, you can say. I have learned a lot from her. So thank you very much for calling me ma'am here and other organizers. Thank you very much. And for coordination, Dr. Indu. So thank you very much. Let me start the topic, today's topic. So you would be seeing that there are two things. Now you type it or draw it. The people who have joined me, they can type it or draw it. See, that's why I asked you guys to try joining me online with the your tool joinpd.com. Still, if you are not able to join, please write IQZAJGH. Start typing, please. So I am waiting for your response. I'll give you 1 minute time. So what do you think? What do you know about this today's topic? Have you heard about this topic before? You can write yes, you can write no, you can write A, B, C, D, whatever you like to. Let's see. Start typing. So I can see. You can directly interact with me in this join PD. See, you can see now. Are you able to see? Yeah, I am seeing the people are writing the response. So 5 students have already responded. I'll show you the response, how it looks like. Some says yes, another someone's next person said. So you can see the responses. Yes, no. Someone has written it and deleted it. So what do you know about it? Statistics. Yes, no. Someone is typing, still typing. So out of 21 students, I have seen 6 responses. Someone says, yes, I have heard about statistics. So I see. So let me hide the responses now. Out of 21, only 6 has responded. So this means only 6 people are with me. 7, 8. So guys, be on your toes because you all only 12 have responded. 13. Out of 70, only 21 were able to log in. That's great. Anyway, let me start the topic, today's topic. So what do you say? What do you understand with this? What is a data collection? So statistics is all about data collection. So you can see data could be in another any form. Who can collect the data? You. You can collect the data. There are various ways to collect the data. Where you can collect the data? There are different places you can collect with your research. There are primary kind of data, there is secondary kind of data. What you can do with that data, I'm going to teach you today's talk. Where you can collect the data, who can collect the data, when can you collect the data, why you have to collect the data, how you have to collect the data. Then what questions you have to ask from that data. Ask the questions from that data, then answer that data. You are you are doing some sequencing, you are doing running abcd in your gel electrophoresis. You are running gel. After running gel, you are getting DNA bands. Those bands you are seeing, you are getting abcd of it. Okay, atgc you are getting. Then you are seeing which gene it is. You are running some tools. So that is question. You are asking question what it is. So then you have to think when you have to collect the data, why you have to collect the data. All researchers, my dear friends, all of you are researchers now. All of you are you are no more BSc students, you are MSc students. It's by choice you have come here. No one has forced you to do MSc. So you should know how to collect the data. How many students have done MSc in Padma Shri? How many students have done in MSc in Bangalore University? So those all collecting of data is called data collections in our. So that's also one kind of research. So you have the arrays here. So what is data? Data could be anything. It could be numbers, measurement that you have collected as a result or observation. You are seeing, today I have given you some observation. Go out and see how many people are there. So you have counted, this many are there. Then uh, someone has given me interesting answer. Sir, there are 10 males were there and 12 females were there. Very good. Someone else has come and observed, sir, there are 10 males, 12 females, and I have found out there was few children also there, few kids also there. So it's a, you are arranging, systematic arrangement is going on. So the systematic arrangement, what is that? And so you are making an observation. So that your result is an observation. So how you are working over it. So the array. So systematic arrangement of your objects, usually in rows and columns. That's called your data arrangement, data array. So data array could be anything. It is your observations that you are arranging systematically. So Data could be two types. You can arrange it in two types. You can arrange it in randomly. You can arrange it sequential. For a science students, when you pick up the sample, you say it's a random sample collection. For a scientist, if you are a business person, then also you can collect it as you wish. So it's like different ways you are going to collect the data. So either it could be random could be 1 7 8 2 3, any could be any number. So sequential arrays, sequential means 1 2 3 4 5 6 7 8, so like that. So those way you are getting the data, that's called sequentials. Now variations and sampling, look into the data here. So you a flower I am showing you here. So the flower is what? Just a sample. It's just a sample. What you are seeing here, you are seeing here sunflower. So you are seeing just a sample here, but I want now a variety of the samples. So what do you mean by the variety of samples? So I need different kinds of flowers here. So when I start with the flowers, look at this. This will become variation. You have different kinds of flower here, so that's become variation. So you are having a different ways of presenting flowers, so different ways of having variations of flowers. So those all you are taking up. So you are taking samples. I said please go and collect the samples. So you started collecting. So what sample? I said okay, flowers, I want flower samples. So you guys started collecting the samples, flower samples. Then after you are collecting the flower samples, I asked you, I want variety of flowers. So you guys go on and collected a variety of samples. So that's called variations of the samples. Then different kinds of variations of sampling. Now you have to think which kind of variation, even human also in a different ways you have to see in the while taking the sampling. So what is the sampling now? So sampling could be random. If I am taking a variation, I am taking, so I am taking from three groups a different sample I am picking up. So I can pick up anything. So it depends on you how variety, how you can want, how you want a variations in your sample. So if you go for it, okay. Let's stop it here. Let's stop for 2 minutes. Okay, can you draw some samples here? Please draw anything on this slide. I want to see how many, what you can think of the sample. Draw some samples here. If you join me, if you're not able to join, please write joinpd.com. Write this code IQZAJH. Draw it. Guys, draw it. Draw anything. I'll show you people what you people are drawing. So don't worry. If you don't want me to reveal everyone, I'll not open it. Do not worry. Please open, try. Please write joinpd.com. Please write joinpd.com. You will be able to join. And please don't give the space. I please write LQZ. Don't space here. It's LQZ, no space AJH. Please write that. Okay. You will able to join. So draw it, please. Let's see what guys you guys are responding to me. So I'll show you now. Someone has written Jesus save us. Yeah, statistics is always like that. We always remember God. That's great. Someone is blank, only they have gone for it. Someone has made something and deleted response. Okay. Some guys are writing A, I. Someone has written dot. Great. That's very great. Yeah, someone is drawing still. Yeah. Please remember, I can tell you the name also who is drawing this or who is writing this. Hi. Hi, thank you. Okay, happy learning. Someone is drawing some flowers. Wow, this is a sample. Yeah, this is dot. Someone has drawn a circle. Yeah, it could be a zero also. Someone has drawn a star. Beautiful. So you can see what you guys are doing. So it's great. Okay. So let's go for next. Okay. Sampling. So what is sampling now? You have a random population, you have a lot of population. From there, you're picking up a population of interest from your side. That's called population of interest. So you can study the sample, you can study the sample in generalized way as you wish. But if you are working for a drug, so we don't want population what we think for. As example, now corona vaccine people are working on. So what they are working on corona vaccine? So they are doing trial in different population. They are thinking the population in different ways. You cannot take only one kind of Caucasians, or you cannot take black people, or you cannot take brown people, or you cannot take one single style of people and you can work out. So you have to think what you are doing. So you have to do random kind of sampling. So is called randomization. Next, so what do you think, how large should my sample be? This question I'll ask you the end of the session if you are able ready to give the answer. Okay, then, so what is the frequency now? Frequency means a number of observation in the classes, listing of classes in their frequency. So distribution is that way. So look into this now. So this is the table. This is one table, 56, 55, 59, 58. So this is the table. So now I have to arrange these 40 employees of an organization. How will I arrange it? Let me arrange it in different ways. So let's see here. So I started arranging it as a classes. So I made it a group 55 to 57. So observation is 55, 56. So what is the frequency? So frequency, what is the frequency? So frequency is 4, 58 to 60, frequency is 6. So this many are there. You can look over your uh, before the numbers are there. How many 56 are there? So we have arranged it in a observation ways. So we have made a classes 55 to 57, 58 to 60. So we have made it in a proper regular interval. So 55 to 57, 4 are there. 58 to 60. So see the writing style here. So how we will write it? So 1, 2, 3, 4, then we cut it. So this is 5. This means it's a 5. Okay. Then 1, 6. So writing observation styles are always like this. So whenever you are doing tomorrow questionnaire surveys, if anyone is management person are here who is here listening to me, if they are doing the observation survey or questionnaires, you have to make it like this. So when you're making like this, then how you can make it look beautiful? So whenever you are going to publish, people may ask you, make it a beautiful way to show your data. So what you do, you will make a graph. Okay, if it would be today workshop, I would have been teaching you uh, Excel, MS Excel, or Prism software, or SPSS. I would have teaching you those all softwares. So there you can write your data, you can make your data and see the structures, how beautifully the graphs looks and what kind of graph it is. I'm going to show you next. Okay, so now you have, so the next slide, or are you able to follow me? Please start dragging. Guys, start dragging. Yeah, the people who are with Jesus, start please dragging. All the goats, please start dragging. So I will understand, do I have to increase my pace or I have to be little bit slow? So you can drag the icon. You can drag down, you have the icons, so you can drag it. Wow, that's great. So I am seeing it. Are you able to follow me? So I have seen 18 people. True, they all are able to follow me. So still two guys are confused. They are outside my true window. So let me see how it is. Let's see. Beautiful. So I am happy. Out of 35, 24, 25 responded. Still 25, all 25 are there in true column. Great. I feel good. So yeah, this makes good. Anyway, let's go for next. So more than 50% are able to follow me because statistics is very tough to understand. I know that. So let's go next. So now so it's a toughest task what I'm going to start now. Type and presentation of your data. How many types of data are there? How to present the data is there. Okay. So now, so data, we already learned it. A set of values recorded or one or more observation units called data. Your fan could be data. Your telephone could be data. Your mobile could be data. Anything could be data. A, B, C, D, E, F, G, H, I, J, K will be data. But it will become a proper data when I say, if I call you, A, B, C, D, get up, you will not know whom I'm calling. But it will give a meaningful data. If I say, Mr. Dr. Indu, please get up. So if I say like that, or Miss Dr. Sandeep, you get up. Or if I say like this, so Sandeep is giving you information. So I am only speaking about information. I am speaking about a proper norm, proper value here. So you have here four types of data here: A, B, C, D. Look into that: qualitative or quantitative. B, discrete or continuous. C, primary or secondary data. D, nominal or ordinal data. If you understand these four types of data, you are clear with your statistics maximum points. So you will not face much problem in future. And I'll assure you, if you know statistics, you can fit into anywhere. You can fit into the toxicological people, you can fit into the marketing people, you can fit into the science people, you can fit into the artistics people, commerce people, anywhere you can fit. And I'll tell you one great thing: if you are from commerce background and you know good Excel, you can fit into clinical research and clinical data management also. If you know the terminology, just you have to know the terminology. If you do one software called SAS, you will be getting a upfront job in maximum of the companies. Even IT companies prefer them. Okay, let's go for next. What is data now? So first one I spoke. I'm going to speak about. I already spoke about the 4 points. So I'm going to speak about the qualitative data, quantitative data. So qualitative, it says numbering, enumerations, attributes. If I say religion, if I say sex, if I say blood groups, so it should be expressed as numbers. What does it mean? But without the unit of measurement. Please be clear in this point. Quantitative means measurement data. As example, blood group, I can say A group, B group, O group. Okay, I can say sex, male, female. I can say religion, Christian, Hindu, Muslim, Sikh. So I can say like that. So there are no measurement. There are no unit. You cannot say Sikhs have some unit. Okay, yeah. If you go in army, Sikh regiment, then it will become a quantitative data. Okay, so you have to understand the qualitative data and quantitative data. So quantitative data is you have a measurement, you have a specific number, you have a specific magnitude. Qualitative data, you can express it, but you don't have any measurement, you don't have any unit of measurement. So please remember this. So qualitative data and quantitative data, if you are understanding, now I'll say quantitative data, I'll say height in centimeter, hemoglobin in gram, percentage, you have BP in mm, weight in kilogram, or I'll say uh, Maggie, how many Maggies you have purchased, numbers. So if I say how much population is there, a number. Okay, so this is the way it's called quantitative data. So so we are doing a long like it's from 30 minutes I am speaking. So let's take a break, a small break. Have take 1 minute of stretch, go one side, this side, that side. I know statistics you are learning, you are already moving here and there, but it's my, after 25 minutes, it's my responsibility. You people have, I have to keep you healthy because online classes are taking a toll on our health. So let's stretch. So guys, let have a stretch. Stretch, guys. Done? All of you are able to do it? Yeah. Yeah. Stretch with me. Yeah. Yes. Yeah. Stretch, please. You can get up. You can move one side. You can come back and sit on your chair. Please. So you can say me hi. So that's also movement. You can say this side. Yeah. You can move this one side, another side. So stretch it, please. So it's a stretch break. Yeah. Yeah. Great. So it's 1 minute over. So thank you very much. Let's go back to our class. Okay. So let me go back to my class now. So now let's go for next presentation, next slide. So now next is discrete or continuous data. So what is the discrete data? You have a whole number. You cannot say half in it. Number of beds in hospitals. How many malaria cases? Continuous data, it could be, value would be possible. You have hemoglobin level 9.8. So you have a point value. You have a floated value. So you can understand in this way. So measurement or possibility of getting fractions. So you have that. So hemoglobin level, height, weight, this all could be thought of. Okay. So hemoglobin in gram, that's become a quantitative data. Qualitative data means how many anemics are there, how many non-anemics are there. Height in centimeters, quantitative data. Qualitative data, tall or short. That's it. BP, BP level, BP in mm of mercury. You say hypertensive or hypotensive. IQ score, genius or normal. As you all guys are genius here. So you all are hearing me, you are understanding, you are able to follow me. I have seen, that's great. Okay, so next, so the types of data. So quantitative, continuous. Quantitative could be continuous or discrete, or it could be categorical. So categorical means ordinal. Ordinal means order, like which grade you are in. I am studying in second standard. I am studying in 10th standard. I am studying in 9th standard. Ordered category. Okay. You are saying which grade of breast cancer you have. 1A, 1B, 2A, 2B. So which grade it is? Is it better? Is it worse? Are you agree with me? Are you disagree with me? If you go for another type, nominal data. It's not an order. You say male or female. You don't have any choices here. You have only sexes. Two types are there, male or female. Yeah, there is another way, another is also there, but usually we don't consider. Now these days they started writing in the forms, but then also male or female, alive or dead. Blood group, you have O, A, B, AB. Someone says, sir, there is RH also there. There are another different kinds of blood group is there. Bombay group is there. So like that, if you see there are out of 26 letters, you will find maximum letters on your blood groups. So that's just to argue. So that you should understand before learning anything. Before you are doing something, you should know what kind of data you are working on. Okay. So now next is, now this is the major task. If you are from commerce background, if you are from management background, even if we are from science background also. So what is the primary data? Primary data means usually science guys do it. They go to the lab, they work, they get precise information. I want to uh, protein estimation. I do Lori's method. I did. We got some proteins. Bradford, These all methods when we do it, okay, biurate method. Some methods we do it and we get the protein data. That is called primary data. Means the first hand information. So what is the secondary data then? Secondary data means I have done it somewhere, but someone else is using that data. They are obtained from outside sources. That's called data obtained from hospital records. You have gone for a survey, you have gone for a census, you have gone for a survey, you want to check it whether how many people are HIV positive from this hospital, from this area. So those hospitals record you have collected and you started writing it. That's called secondary data. Secondary data could be anything. You take some 10 research papers, collect their data, do meta-analysis on it, publish it. It's a great way people do it. And meta-analysis papers are more preferred than your original research because you are questioning them. You're pointing those people. You're asking them, what is the future generation? Uh, what is the controversial data? What is the differences between your data and their data? So those all follow very highly. Okay, so nominal or ordinal data. Now what is a nominal data? It could be Nominal data could be your eyes color, races, sex. Again, ordinal data, categories could be ordered. So you have to have an interval between the two categories. Ranking in the class, examinations. Okay, so now the next comes collections of data. So now next comes collection of data. How will you collect the data now? So collecting data is toughest task in the world. A small mistake can make everything problematic. So you should collect the data very, very carefully and very thoroughly. If you don't have the idea, you should ask your guide. You should ask your boss. You should ask your mentor how the unit of measurement should be defined. Some people do that mistake, means microgram become milligram, milligram become picogram. This mistake happens. So that's totally, it changes. You think drug you are using, instead of microgram you are given milligram. So everything will change. So you should have accurate values, valid values, precise value, and next should be reliable. Your data should be reliable. Collection of data should be reliable. So now sources of collection of data. Different ways are there. From census, you can collect the data. Registration of vital events, you can collect the data. So there are different ways are there to collect the data. So let's see now. So you usually I given you one homework. Okay, I understood you would have got the answer also. So how much you feel, I'll make you remember again. What is the sample size? How large should my sample be? This is the homework I gave you. So hope you got the answer also for this. Now let me know, how was your homework assigned? Okay, usually in your classes, when you take a homework, when you do some homework, how it was assigned? Do you do your homework assignment at right time? If yes, put it green. If no, at right time, then go for a red one. Now it's your choice. I'm giving you a flag. So down you can drag the flag and you can do it. I can see the responses. Oh, I have maximum students are telling they all do it their work at right time. Oh, I am in a genius class then. This, I am proud of your teachers now. Great. That's nice. So your leader must be a star. Yeah, I know. She's a great one. So Dr. Anuradha is always a great. I know that Madam is always do well. So that's, I'm seeing with your responses. Out of 20, all 20 are in the green zone. So she is making it to work well. That's great. Thank you very much. So let me go for next slide. So I already got 50% responses. I already got 22 responses. All are in green zone. Wow, that's nice. So let me go for next. So you can collect the data from Central Bus and Death Registration Act. You can collect the data from sample registration systems. How many are there? You can do collections. Then you can collect the data from disease notifications. How you are getting the data every day from for corona? How many are dying? How many are alive? How many getting recovered from their office record? So there are people working on it. That's called your record, notification of disease record. They are getting data from the hospital records. So every day you are getting your data updated. So they have some vigilance system in their systems, and they have to put how many corona patients has come, how many recovered. So it will automatically get updated in their in WHO profile or government made software. Okay. Then next type of data you can collect it through epidemiological surveillance. So as example, now epidemic, like pandemic is happened. So this work has been done by the epidemiological surveillance. Then surveys, with the surveys I told you already, questionnaires, you make the questionnaires and then you search out and you write it. As example, if I say you how many people use uh, trigonella methi for diabetes or jamun for diabetes, you had gone, jamun means sajamin cumini. Okay, you had gone to the market, and or you are going to the ayurvedic stores, or you are going to your grannies, or you are going to the village. You just checked how many people are eating jamun only to reduce their diabetes. So health examination surveys, this is the major surveys people usually do it. So you can mail the questionnaires also. As example, a questionnaire will be mailed to you, how Dr. Sushil is teaching you, how you understood, have you able to follow Dr. Sushil? So that's also a questionnaire. So with that, we get to know the feedback is a very good options to understand how a person does, and you can always improve your futuristic approaches. Okay, research finding. So you are doing research in your lab. Re- Research is always not always in lab. It could be anywhere. It could be in shop. So market research, what they do, you when you go to the big bazaars, you see there are different kinds of big bazaars are there. Okay, I am telling how it is. Why I am telling different kinds of big bazaar. Big bazaars all are same, but every big bazaar has a different kinds of products kept lying front close to your billing system. Some places it will be kept, you will be kept Kinder Joys, some kids uh, toys, or some socks. Some places, if you go there, lipsticks or cosmetics will be kept there. So this means that place people are using that is majorly. So that's the market survey they did it. That market research they did it. So that's the way we have to follow. So statistics becomes very useful there. Now, now the last topic I'm going to teach you, presentations of data. How to present your data matters a lot. Some people present it in tabular, some present in chart. Okay, so so it should be facilitated. Then that data should be facilitated for your statistical analysis. Okay, so so you have to form a quick impression. If I'll see a research paper, whenever we see the result, we see in the figure format or tabular format, it, it gives me the answer how the data looks like, what the research you are doing. So method of presentations of data. So method of presentation of the data is, the first step is statistical analysis. Presentation of data is tabulation and charts and diagram. Then you can do the statistical analysis. I think next speaker is going to speak how a researcher uh, how a researcher does their statistical analysis, ANOVA, or some uses t-test, chi-square test, regression, correlations, all the things. So where it is useful and how it is useful. Okay, so you can look into that. The So tabular presentation, table must be numbered. Please remember this. It should be self-explanatory. Title, the whatever title you are giving. Data should include the proportions, dominators. Table should not be too large. It's going in pages, pages, pages. It should not be done in that way. Then figures, if you have a comparison. Okay. So I'll just show you how data looks. See, look in that. Malaria cases, 1100. Acute G, 248. Leprosis, uh, leptospirosis, 60. Dengue, total 1308. So this is the way a table should be. But when we do in science, so we put it if in another way, or we will put it here how much error we are getting also. Another kind of, if you go for a frequency distribution table, malaria happened in adults with which species? The plasmodium vivax, 54 adults, 9 children. So this is the way one hospital data is. Then next, if you see the frequency data, so you can see glucose level 120 to 129. You cannot say in one person today 121 is coming, tomorrow 120 will not come. So that's why you have to take a FBS level, fasting blood glucose level, fasting blood sugar level, as in a 120 to 129, like so male 8, females 4. Now you see in the research paper you will find like this. I think next presenter is going to tell you about it. What does it mean? But I'll tell you just this. The first value is your average value, then plus minus. This is standard error. How much error we are taking? So this means the three values what you are taking, you average it, and then you will use it. The average value, whatever you are using, then you do STDV, standard deviation divided by and uh, uh, under root of n. What whatever values will come, that will be given here. Okay, so though that's the way you have to write your data. Then ABCD, whatever you're seeing here, is this is called statistical, whether the data is good or not good. This all I think next speaker is going to speak about. Okay, so charts and diagrams. So next is graphic presentations. We do use to illustrate and clarify the informations. Okay, tables usually present the scientific data. Diagrams will summarize properly. So that's the reason, to make it your research attractive, people do use charts and diagrams. But then also you should know where to use which diagram. The diagram should be easy to understand. It will save you a lot of words. It should be self-explanatory. That's why today, if you whenever you go for any presentation, any paper publication, usually they started asking these days. So in these days, they started asking about the graphical abstract. That will give a gist of your whole research. So then you should know how to work out on these diagrams. So charts and diagram, there are various kind of charts and diagrams are there. Bar diagram, histogram, frequency, cumulative, scatter, line, pi. So where do you use it? How you do use it? The bar diagrams is a widely used. Whenever you do, you just see always is use widely used. Where they use it, there we have the frequency of the data. If you have the frequency of data, uh, um, so how do you use it? On x-axis you use frequency of the data, another axis you use it length. Okay, so the characters. So there are three types of bar diagrams: simple, multiple, and proportional. You can see this data now. So plasmodium vivax. Just now I shown you the data from that table. So total cases 100, falciparum 60, mixed malaria 25. So this is the way this looks in your data. Okay, that's called simple bar diagram. Okay, so what is the bar diagram? So you can see another things is bar diagram is always used for the qualitative type of variables. Please remember this. So this I have taken from one research paper. So you can see the distribution of the blood group here. A, how much is there? B, how much? Where the frequency you will find it. So x-axis will be your x-axis will be your how much percentage it is. So where you'll see the variation that always comes to your y-axis. Okay, so x-axis is your what is the stable data. As example, ABO is stable data. Okay, so frequency can be changed. So that's the reason the changes things comes on y-axis. So x-axis and the height of the bar is equal to the percentage of frequency. So you can, so please remember whenever you're making a bar diagram, you should know why we are making the bar diagram, where we have to make the bar diagram. So multiple bar diagram. So here the multiple means you have the two types of data. I want to put male females data now here. So that's called multiple bar diagram data. Okay. So if sometimes we will meet, then I'll sure teach you how to make this diagram using Excel, or you ask your teachers, they may teach you the Excel sheet how to do it. Okay, they are very good in it. Okay, you have very good teachers with you people, so you can ask them how to draw these diagrams. So it's very easy using the Excel, or using another commercial software is also available like Prism, as well as you have SPSS. Okay, so these all are things could be used. In today Excel has everything incorporated, so you can always use Excel for it. Okay, so next kind of data is pie chart. So 1 minute, next kind of data is component bar chart. So component bar chart means where you have the same section. So there we want to divide the horizontal bar to the 100%, as example like this. So you want to say poor community, from 10% category obtained carbohydrate energy, 10% energy obtained from protein, 80% obtained from the fat. But if you go to the rich community, they do a lot of exercise, they take protein rich diets. So 55% will take the only energy obtained from the fat, but maximum they work out on the proteins. So 30%. So this is the way the proportional bar diagrams comes. Next kind of comes is pie pie chart. So pie chart is a very interesting method. It is for qualitative, but here it's a 360. You have to make it again 100%. You have to from 100%, how many are A, blood group A, how many are B, how many are AB, how many are O. So you can make it as a pie diagram. Next type of diagram is histogram. So there is very people usually ask me like, what is the difference between bar diagram and histogram? So you can see the bar diagram and histogram. Histograms are without gaps. There are no gaps between, and it's for continuous data. Please remember this. This is for continuous data, and it does not have any space in between. Bar diagram has the space in between. So you there are, and we are using here for the class interval 175 to 200 class interval. So okay, and y-axis we plot the frequencies. Already I told you. Okay, there are no gaps in this. Histograms will be used for the intervals. Please remember this. This is the confusion people usually have it. So okay, so histogram, there no space. Okay, distribution studies group according to their heights. So this is the way people usually do it. So next is frequency polygon. I just okay, I'll go for next directly, because I think I have not left out with much time. So I'll directly go for the pie diagram we have studied. I'll just go for the box and uh, whisker plot. So what is the box and whisker plot is? Box and whisker plot we are using only whenever we have the information that is handled by a different kind of audience. So you will change the value to the smallest and then largest. As example, you are having NEET ranking. You have the bicep ranking, biotechnology skill development biotechnology skill development program. Okay, you have that value. What is the scorest lowest people have scored? What is the highest scored people have done it? From there you make the quarantile. So that's there you make the bar graph like box and whisker plot. Okay, so there are lower quartile, median quartile, and upper quartile. So we will say this many people passed NET, SLET. So these people, you want to say in the median value, more than 50% people, 50% who are scoring, they are passing. So median quartile, we have to go for it. So what is the largest value? On the basis of largest value, your medium value will be defined. So you should know this box and whisker plot if you are doing uh, any data value where you are handling the many data values, you are using it. Okay, so this is like I am having the information from audience, from out of you I am asking, okay, give me the feedback. So out of 90 only 20 has given me the feedback, so I will calculate with that how many upper quartile and all the things we can use it. But this this will be used for certain statistics. It will not be used everywhere. Please remember. Okay. So it will be like conclusive research, conclusive plot. Okay. Let's go for summarization. So guys, please write what you have understood till now. Please write it proper. Let me judge myself whether I was able to convey the message or not. This is, you are not judging you, you are judging me. So please write, summarize what you have learned it. At least one line. So someone has responded already. One has responded already about. Yeah, please start writing. So they have started. I understood. Some people have started writing. So I am hiding the response. Okay. So please write it. After 5, we will go for next. So I'll wait for 2 minutes for all of you. So then we will go for next one. 8 has responded already out of 38. You guys are very lucky. You ma'am is doing lot of classes for you from outside. So calling other places people. So you have to utilize this. I think so. And this is your choice. Let people not judge you. You judge yourself. So summarize what you have learned till now. Kindly go ahead with it. 10 guys has responded already. 12. So I am showing the response. 12 people. About data, it's type, different types of diagram. Wow, that's great. I understood, sir. Good session. Understood what sir has taught us till now. Types of data and collections. So you all are writing. That's great. I understood about collection, types of data collection, and the use of. So you can see the all of you are able to see my screen also, correct? Type of data collection, presentation. I understood about the collection of data and its type. It was a good introduction of biostatistics. Great, thank you. Data collections we have learned. I understood that collection, basic concepts. Good session, sir. I don't want to know it's a good session or not. I want to know what you learned it. Okay, thank you very much for your nice writing. You nicely described. It was a good introduction to biostatistics. I understood about the collections of data and its type, presentation, different diagrams, type of data collection. So I'm right. I'm just reading your responses. So thank you very much. Thank you guys. You nicely described. Now, the last. So you have different kinds of. Now you have to tell me what kind of things it is. So you have cumulative exams. You have awakeness states in the class, 3 weeks, 3 weeks. See, I have added every whole type of data in this one graph. So thank you very much. So thank you very much. Now I'll stop it here. So session is open for the question and answer. Okay. So thank you.
Yes,
Yeah, ma'am. You can open the session for questions.
thank you, sir. Thank you so much for this wonderful session. Seriously, it was a very active session, I can say.
Thank you.
You have beautifully explained all the concepts of biostatistics. Thank you, sir. Now I request all the students, if they have any questions, they can discuss with us, sir.
Yeah, please.
Before going to questions, Sushil,
Thank you.
really thank you.
Good morning, ma'am.
It's a good morning. It is a very good session.
Thank you.
I didn't know that you have such a good teaching talent. I thought you are a good researcher.
Thank you.
My interaction with you only with research perspective and other things. I have seen your presentations in Bicep, but this is a real good thing. Taking
Thank you.
students along with you. Maybe we can utilize you and we can conduct one FDP for all the young teachers who want to pursue their PhD. where they can do 3-4 days biostatistics alone, standalone workshop.
Sure.
We can do it together if you are willing to join hands, if you have that freedom in your college, or it is fine for us. We can take you as a resource person and we can do it singly or separately.
Thank you,
It
ma'am.
is
For
up
you,
to
anything I can do, ma'am. No problem. I can talk to them.
Yeah, you talk to them so that we can apply for some agency for the support, or we can directly mention that 2 BICEP institutions together have conducted this workshop, which we are supposed to do it now.
Thank
We
you.
will do it as an FDP program.
Yeah,
Okay,
ma'am.
thank you, Sushil. If you
Made
have
my
any
day, ma'am,
questions,
actually.
yeah.
Thank you very much, ma'am. You made my day. Thank you. Hearing such a nice word from a great giant like Legend, it's great. Like, I'm very happy. Like, for you, I can do anything, ma'am. Thank you very much, ma'am. So now last, I just want to ask them, all the students, can you draw one elephant on this blank slide? This is the last, that's it. After that, we will stop it here. This is the last slide, empty slide. I want everyone to draw an elephant on it. Just for a fun side. Can you draw one elephant on it? Thank you. Don't worry, I will not show this slide to anyone. This is only for me. Okay, then I'll stop the session here if you don't have any questions. Thank you very much. Thank you, ma'am. Thank you, Dr. Anuradha, madam. Thank you very much for calling me here. Thank you for giving me a chance to present front of you and as well as students. I'm very happy to share my knowledge with them. Thank you very much, ma'am. Yeah. Ma'am, shall I stop it?
Yeah.
Thank you, ma'am.
By the way, I would like to inform you our logo also changed, Sushil. You are using old logo only.
Oh, is it, ma'am? I am very sorry.
It is there in our website. You can go and visit. We have changed before you actually.
Oh, is it? Actually, I've taken it from your website only. Maybe that I have cache in my system. So cache memory will be always pick up the old one. Yeah, I've seen there were 2 logo, one P that another.
That is all.
Okay, ma'am. I'll
P
just.
is one which we have.
I'm sorry, ma'am. I'll do that. I'll correct it. Thank you very much. Yeah. Thank you, everyone.
Yes.
Thank you very much. Thank you for sharing my knowledge. Bye, ma'am.
Thank you, sir. Thank you so much. We'll move on to the next session. For the next talk, I would like to invite Dr. Sudipta Kumar Mohanty, uh, Head of the Department Biotechnology and Microbiology at Padma Shri Institute of Management and Sciences, Bengaluru. He has completed his PhD in biotechnology from Acharya Nagarjuna University and also the postdoctoral research in the topic of genome engineering from the National Autonomous University of Mexico. He has published many research articles in various journals. So, for this very little brief introduction, I would like to invite Dr. Sudipta Kumar Mohanty for his talk on role of biostatistics in scientific research. Sir, over to you.
Very good morning to one and all present here. Actually, my friend made it simple that you understand like what a data mean and how to understand a data and what is the importance of data collections. So these are the very important part in statistics. To understand, if we can understand the data, 80% of our job is done, and remaining 20% need to be done by analysis to come to a conclusion. I would like to start with a very famous quote by a Nobel mathematician, A.N. Whitehead. This quote is, he says, the aim of the science is to seek the simplest explanation. I am audible, right? Hello, Madhusri, I am audible,
sir.
right?
Yes, sir, you are audible, sir.
Yeah. So the aim of the science is to seek the simplest explanation of the complex facts and seek simplicity and distrust it. So maybe the statistics does this one. So it presents the complex fact of science and the complex explanation of the science, and it presents to us in a most simpler manner, or the most simplest way it presents, so that we can understand the data in more easiest form or more simpler form. When I just collected, I was seeing many definition of statistics. So this is how I can present to you in the very easiest way. So statistics is the science of learning from the data, and it helps us to use the proper method to collect the data, employ the correct analysis, and effectively present the result. These three things are done in the statistical analysis. So first thing already been done, that how to collect the data, what is a data also you understand. And the next thing is how we can employ a correct method for data analysis, and how we can present the data. These are the two things are remaining. So here, when we listen about statistics at the beginning, generally we hesitate, that we tell that, oh, ye mera bas ki baat nahi hai, this is very difficult for me, I studied a long time back mathematics. All these come into our mind. So statistics in research and academia have rarely been viewed in the positive light, at least at the very beginning. So like always we think that like it involves a lot of mathematics, I should know the derivatives, integrations, or probabilities, or many other things. So we just take a back before going to statistics. But when really we think it is not like that, maybe I will talk a little late about these things. But however, all of us got a relief after a happy marriage of computer science and statistics. So there are a lot of programming, computer science, there are a lot more programming languages available, like Python, this R script or R language, R statistics. Then we have statistical analytical systems, SAS, we have SPSS, we have many other softwares are there. So you just type in Google and you will get a list of softwares in statistics, which is very user friendly. If you don't know, you don't have to remember the formulas. You can go ahead with just type how to calculate correlations. So you just enter your values, it will calculate the correlation within fraction of seconds. And the statistical analytical systems, I have also certified in that, SAS. So it's a very widely used, most commonly used worldwide. So I think starting from the shopping complex, shopping market, means big shopping malls, even computer related companies, softwares, many people use. It's a very beautiful diagram it gives for you, very beautiful phylogenetic tree, it makes many very applicable everywhere. Only thing is that we need to practice it, and it's very fast also. So due to the advent of this kind of technologies, we are like everyone is relaxed. You need not have to remember all the difficult derivatives, or how the formula is being derived, or you don't have to remember the complex formulas. So there is not a single area can be found where statistics is not playing a vital role. Everywhere you can find somewhere, like knowingly or unknowingly, we use every day. When I get up early in the morning, even a uneducated person also use statistics when he gets up in the morning. When I start the day, or when you people get up from the morning, you just calculate that how much time is there for you to get ready, how much time it takes you to reach your office, or how to do your daily work, how much time you have. So unknowingly you are calculating something. That is nothing but the statistics. Okay. So statistics has now been recognized a universally accepted as an essential component of research in every branch of science, not only biology. Starting from agriculture, biology, education, economics, business, management, medical, or engineering, all the fields. And you can see all here I have kept some professions. Like whatever may be the nature of the research and development that involves the application of scientific methods, processes, and systems in order to evaluate and interpret the data. You may be an architect, you may be an astronomer, you may be an author, or you work in a bakery, or a farmer, or a firefighter, a pilot, a plumber. You will be knowingly or unknowingly will be using statistics. And the next question is, why should we use statistics in our research? That is the next question. So like these are the things whatever I have highlighted, I have just mentioned here. So method, we understand a method how to collect the data. That is the very important thing. Like when I talk about, suppose this current pandemic is going on, so some maybe you will be seeing that this vaccine trials are also going on. So how they should understand on whom they should try the vaccine, where they should try, because world population is so vast, each one of us is different. So how they will choose the race, how they will choose the gender, how they will choose the normal or healthy individual, all such a great problem. So first of all, we need to understand on whom they should try and how they will collect the data. So what sort of data need to be collected? That is very important thing in the statistics. Then second thing is uncertainty, errors, and outliers in the data can be avoided. Uncertainty means like if we do in that way, so whether the experiment or whether we can tell that, yeah, we have successfully designed this vaccine. Can we state that one, or our vaccine is this much % efficient? I think I was seeing Pfizer is having 90, today morning I was seeing they are getting the success of 90% success. So what basis they are telling? They cannot give a trial on their company of 10 people, or in a single district they can give a trial and they cannot tell that our vaccine is working at 90% efficiency. So then next thing is errors, like how to avoid the errors. Outliers means which is falling out of the group. Like outliers in simply I can explain you. Let me tell you in a much clearer way. Outliers means suppose in your class you are getting 60%, 70%, 80%, 72%, 75%, like that all people, and one or two students got 98%. So when we'll make an average, so average will come 80% will come the average. The average becomes high because of two outliers. Only 1 or 2% got 98%, or 3 student got 98%. So because of their higher percentage, all average become increased. So those are called as the outliers. If I tell that your average percentage will be 80%, so who has scored 70%, that student will become much happier because his average is become higher because of these outliers. And the outlier, the person who has scored 99% or 98 point something, if I tell that all of your average is 80%, so you will be sad or you will not be happy that my percentage become reduced. So those are the outliers. So how to reduce the outliers in the data? And next is whether to accept or reject a hypothesis. Hypothesis means an assumption, like you are doing certain work, certain experiments, certain research. You are telling that my experiment work or my treatment work or my drug work in a positive way. So you are assuming something. So after that, you will be able to reach a conclusion that whether a particular hypothesis, like whatever you have assumed, is it correct or not, whether to accept that or to reject that. So we'll come to a conclusion. Then the final one is interpretation, summarization, and communication. Interpretation means we will interpret that this is how it happens. So you can give the conclusion. And summarization means you will be summarizing it that because of this treatment, this much of enzyme activity goes high, or because of reducing the temperature, the enzyme activity coming down. All this summarization you can do it, and communication. Finally, how you will communicate to the people, or how you will represent it to the public, that is called as communication. So communication in different methods, like that either you make an interesting graph, you will make an interesting picture, diagrams, or many way of communication in that one. So this is why we use statistics in research. Then next, coming to biostatistics. So I was also searching for the definition for biostatistics, and somehow I convinced in this definition, it is basically the statistics which is extensively used in biological science, or we can tell that applied statistics in biology. These are the three words I can tell. Specifically, biostatistics is the branch of applied statistics that concern the application of statistical methods to medical, genetics, and biological problems, or I can tell this by simply biological problems. So this is a applied part of the statistics which we generally use to address a biological problem. This is how the simple definition of biostatistics. So it is mainly statistics which is extensively used in the biological sciences. So now, how biostatistics helps us? So biostatistical test provides us the means to interpret the data set accurately, so that we can make unbiased decision on how to proceed by knowing the data, what the data says. We especially understand by knowing the what the data is saying. So what the data explains us, we understand by doing certain tests. So biological or biological experiment, or the biological data, whatever the data we have collected, what the data represents to us, or what the data say to us, that's what we understand by performing certain biostatistical tests. So biostatistical test provides us the means to interpret the data set more accurately, so that we can make an unbiased decision on how to proceed by knowing what the data is saying. So first step is we need to understand what the data says to us, like how the values are distributed, how they are far from each other, whether they are from the single population being collected, all those things. That part is known as understanding the data. And after understanding the data, we will do some analysis. And those analyses are of different, again, I will be coming a little late about how to do the analysis. And once we do the analysis, whatever the values we get it, and based on that we take the decision. That is called as interpretation. So we interpret it, and after that we communicate. Fine. So this is how a cycle is. Like first is understanding, you can see, understanding the data. Then designing the experiment to answer the research question or the research problem, like Madam has earlier discussed about how to define a problem in the research, or how to define a research problem. So based on our research problem, we will do the experiments, and in experiments we have always this control and the source of variations and detect the outliers. Then after doing the experiment, we get the data. So we arrange the data that I have not mentioned. So visualize the data and analyze with statistical models. So once we analyze with statistical models, we get a knowledge about the data. So it gives us much more understanding about the experiments, or about the outcome, whatever the outcome we got from the data. It will make us understand more detail about what sort of treatment we have given, how it works, all those things we can understand. So it increases our knowledge. So based on the data, based on the outcome, we interpret practical and statistical significance of the result. We understand the what is the significance of our experiments, based on the treatment whatever we have given, or based on certain advertisement we have given, or based on certain some it's data like with respect to your experiment, whatever you are doing it. And so it makes then once we interpret the data, then what will happen is we like significantly we present like scientifically sound decision we can make it, and we communicate those data with the public, or we present it or publish it, or whatever your goal is. Like you might be understanding, you might be remembering, like whenever you enter into a shopping mall, like some people standing and they tell that you have a like lucky draw coupon, we are drawing it, so you fill your name. So if you'll see very closely that form, in that they will be mentioning what refrigerator in your home it is there, what kind of mobile phone we use. So everywhere they take a data, like or when you are purchasing something in Amazon or some websites, like or some commercial like online websites, they will you will, before buying that, you will see a review, like below that few people have written this product is like this, and some stars will be given. Or when you take a ride with this taxi ride, whenever you take, after you ride, they will take a feedback, and you have to keep on a star, 1 star or 2 star or 3 star. All these kind of some statistics is going on behind that, and based on that they will take the decisions. Like many companies like food, or your some nutritional products, or some juice companies, some chocolate. Now you will enter into any mall, you will be full of some chocolate, some sweets, some different packaging will be there. So before that, they will be assessed that what people want in this festival, what kind of product it is needed in this particular season. Based on that, they will be doing some. So statistics is very important, like to assess, like either like to give an like a commercial benefit, or to get like a profit in a company, or when you are in a like a researcher, like what sort of like a result, based on what sort of result you are getting it, based on that what next experiment, whether you will go for it. If you are going for a particular experiment, then what kind of test you need to do. So all such it will be decided based on that. So like we have generally in biostatistics, generally we do two kinds of tests. The first one is known as descriptive biostatistics. So in descriptive biostatistics, like here we do a parametric test, like methods of producing quantitative summaries of information in biological sciences. So whatever the data we get it, we make a quantitative estimation in that. So here we don't take a decision. Here just we do analysis, and we try to understand like how the data we are getting it. Like how, like I will give an example. Like you might be much aware about the like measures of central tendencies, like mean, median, mode. Mean is like average, or median, or mode. So this comes under descriptive statistics. Descriptive statistics, it gives us a knowledge about the data in general, that our data, how they are distributed, how they are dispersed, whether there is much variation among them or not, or those data are originated from how many, what is the population size, what is the sample size, how many samples, whether it is replicated or not. All such things it will give an idea. So like how we represent, once we get these parameters like dispersion, standard deviations, the standard error, or measure of variations, all these comes under descriptive statistics. So once we get those values, then we need to present them in a tabular form. Either you can see a table, like this is a statistical table. So there you will have n is the what is the number of like this is the population size, like sample size 115, then something is 65 or something, then m is median, standard deviation, standard error. So getting calculation of all this, we will understand that okay, this is the suitable for next level or taking proceeding further. So it gives an idea that our experiment, whether it is giving a result based on our assumption, based on our hypothesis, or based on whatever we have assumed earlier. So in descriptive statistics, we use certain tests like measures of central tendencies, this average,
Sir?
median, mode, and all.
Sir?
And hello.
Sir, sorry to interrupt. Sir, your screen is not properly visible, sir. Only like a green colored thing we can see.
Screen is not visible.
Now it is visible, sir. Yes.
Okay.
Okay, sir.
So descriptive statistics, it gives us these are the things, like it gives all the parameters. We just analyze these basic parameters, especially quantitative parameters, mean, median, modes, measures of dispersion, standard error, standard deviation, all this. So by looking into this, we can represent them in beautiful graphs like bar diagram, bar charts, or which will be easy to understand. Rather than reading all this data, you can see these are, when we see the table, I have to read each and individual data. So if I will present in a graph, like in a color format or some pie chart, bar diagram or histogram or something, I can easily understand, okay, this red part is this much, this green part is lesser than that, orange part is something very less than that one, and the smallest part is the light blue color or something like that. So coming to a conclusion will be much easier in the descriptive statistics. And next one is inferential biostatistics. So inferential biostatistics, here we come to a conclusion, or it helps us to take a decision that, okay, now we can go ahead in this way. So this is the method which makes us a generalization to take a conclusion like about the larger group based on the information about a sample of that group in biological sciences. Means we test, suppose we conduct a drug test on small samples, like at individual state or different state, we will take the sample and we will conduct the survey, or we conduct the trials of a particular drug. So once we complete, we take the data, and after that we analyze by preliminary. It is the inferential statistics. It is done in two ways. One is known as estimation, and another one is known as testing of hypothesis. So we test them using a hypothesis. Hypothesis, again, I told this is an assumption. So there are various hypotheses, various tests are there to test the hypothesis. Like we have chi-square test, we have your ANOVA, which is most commonly used by the biology people or the biological sciences. In medical, agriculture sciences, ANOVA is the most widely used test to confirm the significance level, or to test how our result is significant, all this. So inferential biostatistics helps us to make a decision about our treatment in the experiments. This is the workflow I will tell in the biostatistics analysis when we do for our experiment. Like if the first step is we collect the data, and then next is the presentation of the collected data, or the arrangement I will tell. We can arrange sequentially or how we want based on the arrangement and presentation of the collected data. Then the next one is analysis and interpretation of the result. So either we have to go with the parametric test like the descriptive statistics, and next one we can go for if we want to come to a conclusion that simple thing, suppose by giving such kind of, or increasing the temperature or decreasing the temperature, or increasing the pH or decreasing the pH, whether increasing or decreasing or variation of such kind of parameters have any influence on the result. If we want to come to a conclusion, then we will go for the inferential statistics. So inferential statistics, I told. So basically it is done by testing the hypothesis. Like more details, since we don't have the time, maybe we can go more detail in this, how to formulate the hypothesis, how to test the hypothesis, and how to find the significant level of the treatment, like what are the parameters we are testing. Maybe sometime we'll have much detailed discussion, or maybe how actually it is done, I can explain. So the last step is making based on the inferential statistics, we make the decision on the basis of the result we get from the test. And some examples I have kept here, to find the action of a drug, or a drug is given to animals or human being, to see whether there is changes produced are due to the drug or by chance, or to compare the action of two different drugs or to successive doses of the same drug. There are many examples, like not only, suppose one advertisement is given, because of the advertisement, whether the sales have gone increased of that particular product or not, whenever the season changes, like you can see, whenever summer comes, people the company producing more like cold drinks or juices or ice creams, all that. So someone is looking behind at the when very simple thing of understanding is that whenever you are entering into a crowded place or a market or shopping complex, you will be seeing while purchasing billing counter, you need to enter your number. So based on the number, they will analyze that how many times you visit that particular shop or particular mall. If you are visiting, what kind of purchase you are doing. So based on that, they have different plans, like club customers, you are some different row they will keep, some special privilege they will give. All those things are going behind. So role of biostatistician, I will tell you, we need to identify and develop the treatment for the disease and estimate their effects, like in regular day-to-day experiments what we do, and identify the risk factor for the diseases, design, monitor, analyze, interpret and report the result, develop statistical methods to address the questions which are arising from our research, and the ultimate objective is to improve the quality of the research. These are the role of a statistician, especially biostatistician. Whenever I will talk with respect to who are a researcher in the life science, so these are the roles and responsibilities. And in conclusion, I will tell statistical techniques can assure that the results found in the study are not merely because of chance. So it should be repeatable, like whatever the conclusion or the inference you will draw, if somebody, if he will do also, it will also happen in the same way, or that is what we give the standard error, plus minus 2, plus minus 0.2. So closely it will follow. The trend will follow in the similar way. That is what the conclusion is. That means it will assure us that the result, whatever you are coming to in the conclusion of your experiment, whatever the result you got in your research is not because of the chance, because of certain impact of your treatment it happens. That is what you want to assure or confirm it. So a well-designed and properly conducted study is a basic prerequisite to arrive at valid conclusions. So you need to design your experiment properly, or for conducting the study is the very prerequisite to arrive at the valid conclusion. Thank you very much. And we have also a activity. So there we'll tell you though we have different softwares, but here I would like to teach you or getting familiar with Excel. Like there are also many functions are there. Excel is a very powerful tool which is known as pivot table. So there it's very fast and it's a very powerful tool by Excel. So I will make you familiar with that. Even my friend is also there, Pinon. So we'll make you a practice in this, like taking a data set, how to arrange the data, how to sort the data, how to make certain analysis like the standard deviation, standard errors, all this parametric test, descriptive statistics in just a couple of will take maybe half an hour. So after this, you make ready your laptop or computers. So we will be with you after a short while. Thank you very much.
Thank you, sir. Thank you so much for this nice session. Students, if you have any discussions, you can. As sir told, there will be an activity session soon.
Shall I stop the screen, right? I will close this screen. So
Yes,
we'll
sir, you
be
can
starting
stop.
with the activities, right?
Yes.
Hello, Preenon. You can unmute Preenon. Akshay, can you unmute Preenon?
Yes, sir, I've done it, sir. Sir,
Yeah.
two IDs have logged in.
Hello.
Preenon.
Yes.
Yeah, you can start. You can start. You just tell, like opening an Excel and how to add in the analysis, like data analysis plugin into that. Yeah.
I will teach basic statistics so that
Okay,
it will
okay,
be
fine.
easy for them for any work. Basic, very like this one, mean median mode, then bar
Yeah.
graph, pie chart, and so on.
Okay, fine. I am also there. And
Yes.
how many of you are having laptop? Can you raise your hand or we want to see that? What is the number or total number? Yeah. How many of you are having laptop currently with you?
Anybody is having laptops or they are using their mobile only? In mobile,
That's what we want
it
to
will be
know.
very
How many
difficult
of
for
you
them to
are having
understand.
laptop? Can you raise your hand?
Yes.
Let us see just how many of them are having laptop or mobile phone. Mobile phone also Excel can be like installed, right?
No, mobile phone, it is difficult in mobile phone to do.
I
I don't know if anybody is experienced in mobile phone in Excel is okay, but I
Okay.
cannot do in a mobile.
No,
Yeah.
your thing, from students I'm telling. Students, how many of them are having the
Swati,
laptop?
Anjali, and Uma madam. Yashwanti, Yashwanti doesn't have. Uma madam, Anjali, and Swati. 3 persons. 3 persons have only told that they have Jovita, yes. What to do? Shall I start, or because I think 4 persons already told that they have a laptop. Swati, Anjali, Uma madam, and Jovita.
Okay. I would like to tell you all students, like since this is the age of what I can tell, computer. So we don't expect that a very costly computer. At least you buy a machine like which will cost you to perform very basic analysis, like either you will online writing assignment, this, that, and for everything you need a computer, a laptop. I think you purchase a laptop so that it will be much easier, like writing, doing your assignment or any other thing is much useful in all the things. So that you just this is one of my opinion. So those who are not having, I expected that many of they are having the laptop, but here I could see only 2 or 3 or something. Maybe other people can see because here the statistics, it is like if you do by your own, or if you operate by your own, so it will be like you will be getting like much handy for you. So you'll be getting more knowledge if you'll be doing by yourself rather than seeing that one. Anyway, you can start. You can start. We can. Yeah. So
Yeah.
you tell from the beginning.
Okay.
Yeah.
Yeah, I will start very basic. First, I will start with the mean. Before that, I will start with the summation. So, for
Yeah.
example,
Fine.
if
Fine.
there are numbers like this written, yeah. So just if you have to sum these numbers, it is very simple. Just select it. Main thing in all the statistical operations in Excel is you have to select all in this manner. Leave one space. Then you can see there is an auto sum. So do this auto sum. You will get this value. So this is what if you are removing this, like as I told you, this is the sum value. If you are not selecting this column and you are selecting in this manner, you will see what happens. Yeah, this is taking other things, it will not take. It will come in the overlap in this only. Okay, so average is there. Average is the mean. Yeah. Okay, I did a little mistake. Okay. This is the average. Average means sum of all the numbers divided by, or dividing the how many numbers are here. Average is, I think everybody knows it. This is sum of all the numbers divided by numbers present. So this is your average as well as this is your mean. Those who are having the laptops, they can just do it accordingly. Those who are having the numbers, just you can do it. Just randomly you can type any numbers. Randomly you can type any numbers and you can see if you can get the auto sum and average. Once if everybody is doing it, I will proceed with the next part. And just put it in the chat box that yes, it is done. Okay, one person, Jovita. Others? Those who are using the laptop, please do along with me. It will be easy. Only one person. Others? Is my voice clear to all?
Yes. Yes. We can. Yeah. We can listen.
Okay.
Go ahead.
Okay. Okay. So 2 persons are telling that they have done it. Others? Okay. Those who are having statistics also, for them also it is easy because now in the Bangalore University syllabus also this is included. Okay, I think everybody has done. So I will move to the next part. Okay, so select all. There is an option here like more functions, statistical. Then you can get it here like average is there, so many parties there. I will go for the mode. Yeah, mode is here. Yeah. So what is mode? I think every people know it. The most value. So here, this is the answer means how many times mode is there? 12 is there. 1, 2, 3, 4. So 4 times 12 is there. So this is your mode. Everybody, please do it on your own. Let me see whoever is getting it. Everybody can do on their own and see if it is for them you are getting or not. Yes or no? Is everybody able to do this mode? I don't know. I don't see any response in the chat box. Nobody is able to do this. Do I need to repeat, Sujita sir? Nobody has
No, we are
responded.
able to listen. Yeah, you tell.
Okay.
Like, yeah, we
Yeah,
are
nobody
listening.
is
You go ahead.
doing it. I don't know.
Okay, please respond in your chat box that whether you are following him or you want to
I
repeat or
don't
what
have
you are
mode
doing
function.
updated.
I don't have mode function. Why? It has to be there. It is like, I don't know. It will be there in statistics. Statistical, then you will have a mode. Here you have almost all the statistical function you will have.
Hmm.
So two mode functions are there. Mode single and mode multi. I think mode single. Mode single should be the one. I have only one mode. So Madan, are you getting it? In mode single should be the one. Yeah, Anjali has responded like she is getting it. Others? Okay. Yeah, Uma madam, it will be there. Basic mathematics and statistics will be there. Okay, anyway, I will move to the next part. One more is like median. Okay, let me select this. Okay, so let us select this numbers only, and there is function like statistical median. That's all. Yeah, here it will be because the number is median is the this one middle value. The thing is that if you are told I am taking this series, it will be 12. So let me change it. Okay, I just have given a normal series 1 to 8. Let's see what is the answer coming now. Yeah, 4.5. Okay, great. So mode is between this both numbers, sorry, median. So this average of these two. For even numbers, it is it will be your n by 2 and the sorry average of uh, no, it will not be like, it will be like n plus 1 by 2. That is equal to 8 plus 1 by 2. That is equal to 9 by 2. That is equal to 4.5. Very good. Suppose you had these numbers 7. Somebody has raised the hand. 1 minute, I will come to that. Yes, who has raised the hand? Somebody has raised the hand, Akshay sir. Sudipta sir, somebody has raised the hand, it seems. I cannot see who are they. Divya and
Divya
Madan.
Verma and Madan. Yeah.
Yeah, Madan. Yeah, you can unmute, I think. Can you unmute yourself or you can just leave the chat box?
Either way is fine, sir. I've asked them to unmute. They could either unmute or they can use the chat.
Yeah, okay, yeah, tell me you have raised the hand. Any doubt? Okay, Madan, no problem. Divya? Divya? Divya, you can unmute or you can even leave a message.
Sir, I guess it's by mistake, sir.
Okay. Okay, no problem. Oh, Madan by mistake. Divya has not responded. Okay. Okay, fine. So this much I am doing it. Then there is option for sorting. So sorting option was here. Sorting, I think you have to select these numbers and yes, smallest to largest and largest to smallest. So you can see here, what is this 2.5? Why it has come like this? Okay. Yeah, it has come like this. You can see here, it has sorted. Then largest to smallest. Everybody can do it. This one also sorting. These are the basic functions, whatever I am taking for you. Then I will come to the pie chart and bar graph. Okay, yeah. Others, Divya is still, her hand is raised. Divya, can you tell me any problem or we'll move forward? Divya's hand is still raised. Okay. Okay, everybody is getting it, how to do it? The statistics students who are for whom I am teaching, they can also, I don't know if they are BSc students or not there, I think. Yeah, BSc are not here. Okay.
No, here PG.
Yeah, PG. Yeah, BSc I am taking statistics, but they are not here. For them, I had to take this part. Okay. Okay, so this is done. Then I will move to the next part. One very simple thing in statistics, you can do it. Like if you want a series, like 1, then it should be 2 should come immediately or 3 should come next. How to do it? Just select it. Sorry. Yeah. So completely it has come 1. Now you can click here and fill series. So 1 to 11 has come. This is this will be very useful for you for the statistics. Any small things. If you are doing it, you can directly get this serial instead of typing it. Just you can type one example. I will show you again. Suppose you are typing one, then you need like 2, 3, 4, 5, and so on. So no need to type like here 2, then 3, then 4. No need to type it. Just what you can do is select here. You will get this plus sign like this. So drag it. Drag it. Say for example, I drag till 15. It has come only one. Only one it has come. There is, you will see one option like a drag down menu will be there. Then you'll have this fill series. Don't give the copy cell because copy cell is default. Then you have to put it like fill series. Fill series means it will come in the form of series. Any doubt? Everybody try to do this. This will be very useful. It is beyond statistics. This part is beyond statistics. Everybody please do it. Yeah, please do it. With Excel, we can do so many things. Even there are books coming for Excel. Because in Excel, you can do so many. We can see here financial, logical, text, then mathematics, and even more functions are there. So more function you can have many. Like statistical is there, engineering is the cube, and other information is there. So many things you can do in statistics in Excel. And above all, I won't say Excel is free, but people can, everybody can get it in free also. The crack versions, it is very easy to get. The other softwares are there, like SPSS is not free, MATLAB is not free. These are others are all paid software. Okay, good. So for these numbers, I will not take this many numbers. Suppose okay. So now what I am going to do is, yeah, I will show you this bar graph. Let us see how it comes. So this is not bar graph. This is your. This is bar graph only. Yes. Yes. Huh? Why it has come like this? Why it has come all.
Select one more
all
parameters.
That right about a single also it should come.
One,
That I did it, but here why it did not come?
one more parameter you enter in another column. Yeah.
Some mistake is there. I think this will come in a revert. Something is I did mistake. Why?